Thanks everybody for coming to our first OCB Metabarcoding Working Group webinar. This is the intention is that this is going to be a bi-monthly webinar series. So we are looking at having our next one in June. The focus of that will be about intercalibration activities, and we're hoping to announce specifics about that soon. Also, thank you to everyone that has applied to be a participant in the intercalibration activity itself. We received a pretty large number of applicants for that activity, and the organizers are currently evaluating those applicants. We hope to be in touch about that soon. So today we have two speakers, Dr. Felix Milke from University of Oldenburg and Dr. Mahwash Jamy from Uppsala University. I think Felix is going to go first, if you'd like to share your screen.
Yes, thank you very much. Okay, does it work for you?
Yeah, looks good.
Great. All right, yeah, thank you very much for the invitation to contribute to this webinar. Yeah, my name is Felix Milke. I'm a postdoc in the working group of biogeochemical ocean modeling here at the University of Oldenburg in Germany. And before I want to explain you what actually microbial cohorts are, as you can see, the title Biogeography of Microbial Cohorts in the Global Oceans, I want you to have a short look at these beautiful network diagrams. So I think, like, for probably most of the people that did this kind of analysis, just the beauty of these figures probably was the main motivator to run these analyses. Because, like, when we look at these figures, it is first and foremost quite difficult to really get an ecological interpretation of co-occurrence network analysis. And I think the most valuable one is the existence of modules or clusters in these networks. And this is quite interesting as we can find these clusters, these modules, in all kinds of different biomes. So wherever we look, we see that the co-occurrence networks microbiome data is modular, and modular is actually a measure. So it's kind of an index or a property of a network that can range from 0 to 1. From 0 meaning that it lacks any kind of modular structure or clusters, whereas 1 means it has quite strong clustering within the network topology. So the question is, why are actually co-occurrence networks modular, right? So because we have this gradient between, on the right-hand side, what we can say or call hairballs, and on the left-hand side, what we can see in reality. These hairballs is usually what we might also say as random expectation, because this is what we end up when we just take the same number of nodes and randomly let them rewire. But even though this would be the random expectations, the reality looks completely different. So there must be some kind of process that structures these networks to become so modular. And this process is environmental filtering. So just to give you an example, here on the right-hand side, we see a cruise across the Central Pacific Ocean. And when we sample or look at the communities in the warm region, we find completely different organisms as in those samples in the cold region. So that means the organisms in the cold region will never co-occur with the organisms in the warm region. So that means in a network, they will never be associated with the same cluster or have an association between each other. So environmental filtering is actually the force that creates these close clusters in these network topologies. And in the ongoing talk, I will talk in more detail about these clusters. And I will not only call them clusters. Unfortunately, there's still some loose terminology here, but I will try to keep calling them microbial cohorts, okay? Because this is what we named them in a recent article. And these microbial cohorts They are groups of prokaryotes actually that share the same environmental preferences. And in our article that we just published, we were looking not only in the ocean, but we were looking at all different kinds of biomes that we could get data from. We had human secretion, we had distal gut microbiomes, we had soil, plant-associated microbiomes, of course, marine, freshwater, indoor surface, and human skin. And wherever we were looking, we always found that the modularity in the networks was significantly more modular than by random expectation. And we can see, or we can describe these cohorts that we could identify in these biomes according to their preference properties. For example, how many ASVs could be assigned to a single cohort, or also how many cohorts coexisted in the same sample. So, for example, the rough order of ASVs in a cohort was about 100 ASVs. And we could find between 2 and 8 different cohorts per sample. And also, in all of these samples, the amplicon samples, we could map more than 70% of the total counts to these cohorts. So indicating that they must be really a significant part of the community. And you can see here that I differed between 2 different correlation thresholds, which I don't want to go into detail much, but if you are interested, I would recommend you to have a look at the article that we just published. So we came up with the conclusion that microbial cohorts are actually universal features in microbial communities. But why is it actually so important for us to know about that? So, for example, when we look at recent articles that were assigned or associating environmental stress with network properties, they found that stress has an effect on the networks and probably destabilizes the network or is somehow negatively associated to the network. But as I told you before, it's really difficult to interpret network properties from an ecological point of view. So when we look at, for example, a data set, which is here in this case, the ocean latitude data set. And on the top figure, you can find a lot of different bars. Each bar represents the community or one sample in this data set. And the color of the color is coded according to the cohort that we identified in this data set. And you can see now that when we order the samples according to the temperature gradient, that towards the end of the gradient on the left and right side, there are much fewer cohorts dominating as compared to the center area, the central area. And this translates, when we look at the local modularity in these subgraphs, this translates in a decline in modularity, in the network modularity towards the end of this gradient. So that means when we say that stress destabilizes microbial network, it looks like that just means that there are just fewer cohorts that are adapted to these extreme conditions and that prevail under these conditions. And this is not only true for the ocean, but we can find the same also for freshwater, for example, here in the Lake Mendota time series, but also in a large continental scale Australian soil data set. So another benefit of these cohorts is that they are extremely deterministic. So the composition of cohorts follows environmental conditions. For example, we can say that the ratio between cohort 2 and 10 in this example here has a linear dependence on temperature. And when we, for example, look at the network that is behind these cohorts, we see that on the left-hand side, cohort 2 and 10, they form distinct clusters in this network. And on the right-hand side, this is the same network. We just color code each point, so each ASV in this network, according to its preferred environmental condition. And you see that these ASVs, they share similar environmental preferences, and that there is a very distinct temperature signature in these clusters. So also this the beauty in this approach lies also that we don't have to look, when we use this deterministic behavior of these cohorts, we don't have to look at hundreds of different ASVs, but instead we can reduce the complexity of microbial communities to few cohorts. And so I try to apply this concept, this deterministic feature of these cohorts in order to globally predict the composition of cohorts in the global oceans. And for that, I first of all had to use another data set, because my data set was only two latitudinal transects, one in the Central Pacific Ocean, the other one in the Central Atlantic Ocean. But the group of Jet Furman and others, they assembled this really great data set, they call the GRUMP data set, that includes much, much more samples. And what I did first is I recomputed the co-occurrence network. So on the left-hand side, you can see this new co-occurrence network, which, well, now we end up with fewer microbial cohorts, fewer groups, but much more points. So that actually, in the end, this doubled the amount of ASVs up to more than 2,500 ASVs that ended up in this network, and that we can now use in order to map cohort composition. And the reason for the reduction of cohorts, like the total number of microbial cohorts, likely lies in the fact that some of these cohorts are merged together, because it's quite difficult to find distinct limits or distinct groups when there are, like, so to say, overlapping cluster borders. So I used this data set and were mapping the cohorts that we identified in this network against the samples. And you can see here we have two different, I subsetted the data set into two different oceans. On the left-hand side, we see the Atlantic Ocean. On the right-hand side, the Pacific Ocean. And there are three different depth groups. One is below 100 meters, then between 100 and 350 meters, and more than 350 meters. And this is what it looks like, the composition of cohorts in each of these samples. So we have this purple cluster 1, that is the warm water cohort, then the gray one, which is the intermediate temperature cohort, and then there's one which is associated to cold temperatures, which is here cohort number 4. And then there's also one interesting cohort, that's cohort number 6, that's the salmon colored one here that you can see in the figure. And this is associated only to the Pacific equatorial upwelling region. So there was only at this upwelling region we found this very distinct group of organisms that only prevailed there. And now I developed, together with others, this cohort prediction framework, which I don't want to go into the methodological details, but we could predict, just based on environmental conditions, the composition of these cohorts only based on temperature, salinity, oxygen, phosphate, nitrate, and silicate. And we were actually quite puzzled to see that there is really great similarity, or a great performance of this prediction. So when we look at the dissimilarity between the observed and the predicted microbial cohort composition, we see that this Bray-Curtis dissimilarity lies at around 0.12, which is really low. And we were even looking in more detail about the performance of this prediction when we were correlating the predicted abundance versus the observed abundance for each cohort individually, and each point here is one sample. And you can see that the linear model behind it really lies on the one-to-one line. And we end up with adjusted R-square value for these linear models between 0.8 and 0.9. And even when we subsample the data set into a test and a training data set, and reduce the amount of samples used for training to only 30%, this framework still resulted in a Bray-Curtis dissimilarity between observed and predicted at about 0.14, so only marginally lower, and really showed us that this idea seems to work really well. So now we used this model, this modeling framework, in order to predict not only the GRAM data set, but also to predict the global ocean. And this is what we came up with. So you can see here an animation of each of these 6 cohorts that we identified. And this is a small animation that is for every 10 days throughout the year 2021 and for 100-meter depth. So we can, of course, also look at other depths, but just for visual purposes, I stick here now to this 100-meter depth. And you can see again, so we have this warm water cohort or cluster 1, then the intermediate water cohort, cluster 2, and the cold water cohort is cluster 4. And then there's also one, for example, here, cluster 3, which seems to be a cohort associated to the equatorial, to the western continental slope, probably due to upwelling, and was really interesting to see that there is also this, interesting blue one. But there is also then again this cluster 6 that you can see on the bottom right, which is the one that we really have difficulties actually predicting, because this is the one that we in the data saw only at the Pacific equatorial upwelling region. And so, but we can now also zoom inside, because this was on a global perspective, and now let's zoom inside and we can see here the southern tip of South Africa. And you can see that this is an interesting region because there's the Agulhas current transporting warm water and the mixing with the Benguela current that transports cold water, and they create these huge eddies. And you can really see that these eddies, we can translate the environmental fluctuation into cohort composition. So it was really interesting to see for us that these cohorts form and also resemble these eddy structures. And when we also look into the literature, we could see that that was already found in 2010 by the group of Federico Balta. And they also said that, of course, there's that eddies have an effect on the composition of microbial communities. And we can now even see and translate these differences into the composition of cohorts. And you can see also in the top map, there are small red points. And these red points are sampling locations in the GRAM data set. And on the bottom right, you can see the observed differences, and on the right, the predicted differences in the cohort composition. And in the observed cohort composition, we find even a higher variability as in the predicted, even though all in all, that's already quite a good match. So, but this is not all we can do with these cohorts. Because like in the initial publication 2023, we also found that cohorts correlate, or the number of coexisting cohorts in a sample correlates with the ASV richness. So that means the more cohorts are in one sample, the higher the richness is. And when we assume that we can predict the richness of a cohort based on its relative abundance, we would end up with something like this here on the right-hand side, where we can really find, so to say, a saturating curve that saturates the richness of each cohort relative to its relative abundance. And now when we can predict the richness of a cohort just based on its relative abundance, and we can predict the global composition of cohorts, we can also predict the total richness of a community. And this is, for example, what we did here. On the left-hand side, you can see on the upper picture, this is for the Atlantic Ocean, and on the bottom, this is for the Pacific Ocean. And this is on the y-axis richness and on the x-axis latitude. And even though that works quite well in terms of order of magnitude, there's still some variation that we just don't cover with this approach. And interesting was also that, for example, in the North Pacific region, there's a huge discrepancy also between samples that we observed and the predicted richness. And this is, I'm not sure, but the upper part of these points refers to gradients 2 cruise and the lower parts to gradient 3 cruise. And there's a huge difference between gradients 2 and gradients 3 cruises that are in the GRAM dataset, even though they were taken at the same location. And I'm not sure whether this is maybe just an issue of using richness as diversity metric. However, I think it makes sense to just not always use richness, but maybe also look at others, like, for example, the Shannon diversity. And this is also what we did here. So we can really see the Shannon values of cohorts composition, how they change throughout the year. So this is, again, a small animation that tells us how the diversity, the global diversity of prokaryotes changes throughout the year. And this looks really promising for the ocean, but we can apply the same framework also, for example, to soil data sets. And this here, for example, is the LUCAS soil data sets. LUCAS stands for Land Use and Coverage Area Frame Survey dataset. And this is a huge data set, more than 800 samples taken all over Europe using the V4 to V5 primer set. And I did the same kind of co-occurrence analysis and also prediction. And you can see that these cohorts that you identified, they assemble strongly across the pH gradient. And the prediction and the observations also match quite well. So to sum this up, we came up with, in our article, a recent article, with this analogy. So because everybody knows the concept of biological organization, that, for example, we start with a cell and multiple cells form tissue, different tissues form organs, different organs and organism into population and into community. And analog to this, there's also the idea, or that we invented this idea, or not invented, but adapted this idea, where we also start with a cell in the microbiological sense. But instead of tissue, we have populations of single clonal organisms. And multiple of these populations can form a functional consortium. So a functional consortium is meant to be a group of organisms that maximize a certain function. And this is analog, for example, to an organ in metazoan. And multiple of these functional consortia is what we find in the environment assembled into microbial cohorts. So these cohorts is actually what we can track in the environment, and where we can really say, okay, so this seems to be a kind of encapsulated group of organisms that together maintain each other's survival. And which is why we say this might be analog to the level of an organism in metazoan, because an organism is, in the end, the level where you have this biological autonomy to really interact with the environment. And all in all, multiple microbial cohorts form the community. Now, these cohorts is really, the concept of cohorts is really great to dissect between community variation within and between cohorts. And we assume that there is a variation within cohorts. So that means a cohort is not always stable, but there can be a fluctuation within a cohort when you look at different samples. But this variation within cohorts is mostly seemingly stochastic. And we assume that it's mostly driven by biotic interactions. Whereas when we look at variation between cohorts, this seems to be very deterministic, or at least we can use this variation between cohorts to really predict their composition. And we are now working, I'm working with Samuel Hürten. He's a PhD student in our group together, who is preparing a huge MAC database and mapping them against metagenomes to infer also the functional information of cohorts. So we try to build the same kind of idea of computing co-occurrence networks, and then try to identify which MACs belong to which cohort, and then identify functional guilds within these cohorts, try to infer genomic properties of cohort members, but also look at function biogeochemistry matches. And beyond that, as a kind of perspective of what will come after our current work, is that I'm working together with Sinika Lennartz, who is also the head of my group, of our group here, Biogeochemical Ocean Modeling. And she invented this microbial community model, where we can really try to go on a theoretical level and try to identify ecological drivers that might yeah, kind of lead to this formation of cohorts, but also have a look at the impact and the role of metabolic exchanges and how they kind of support the existence of microbial cohorts. Yeah, with that, I would like to end my talk. And again, special thanks to my group, to the group Biogeography Ocean Modeling, and of course to Sinika and to Samuel. Thank you.
That was really great. Thank you, Felix. I think we just have just a few minutes for questions if anybody has a question. I see a hand raised from Victoria.
Hi. Hi, Felix. I don't know if you can hear me.
Yeah, I can hear you.
Okay, thank you very much for your excellent presentation. I found it very insightful.
Thanks.
I have a question regarding temperature-driven changes in microbial cohorts. So I was wondering, are there identifiable temperature thresholds beyond which microbial cohort composition like shift abruptly rather than gradually? Do you have like identifiable temperature thresholds beyond which your microbial composition shifts abruptly rather than gradually?
Yeah, this is a great question. And so far, it seems to be that the shifts in composition is really gradually. So you can really say that, that along the spectrum that we sampled between 0 and nearly 35 degrees Celsius, we had a continuous change in the cohort composition. So that means, for example, when you go towards the warmer temperatures, so there's, so to say, the warm water cohort dominates more and more and more in relative abundance. But there was no abrupt change that led to a completely different composition.
Okay, thank you. Cool. Thank you for the clarification.
You're welcome.
I think we have time for one more, maybe quick question. Jed?
Hi, Felix. That was absolutely fantastic. I loved it. I'm curious how much of this is driven by the prokaryotes versus the eukaryotes in the GRUMP dataset.
Yeah, great question. So, like the whole modeling framework is relying on log abundance ratios of these organisms. So we compute ratio of cohort 1 against cohort 2, for example, and that is in this sense quite valuable as it kind of avoids compositionality biases. So, which is why we don't have to, for example, verify the data beforehand or something like that. And at least that reduces the amount or the impact of, for example, the having yeah, certain eukaryotes that take over a large part of the community. I mean, the GRUMP data set is in this sense quite valuable as it includes both the 16S and the 18S. So that means a single sample, when we look at the count distribution, there's always a certain amount of counts that is associated to eukaryotes compared to prokaryotes. But in our approach, we were only looking at the 16S level, so only at prokaryotes. Most of the reason is, so we also tried it for eukaryotes, but the problem with eukaryotes is they seem to be much more stochastically driven, or at least it's really difficult to get this kind of yeah, clear biogeographical patterns that we can get with the prokaryotes.
Thank you. That's wonderful. Thanks.
You're welcome.
Okay, I think in the interest of time, we should move on to our next speaker, Mahwash.
Yes.
Hey.
We can see your slides.
Yeah, okay. Great, excellent. Okay, and I don't see anyone, which is a bit strange, but okay, well, if it works. Okay, so hello everyone. My name is Mahwash and I'm a postdoc based at Uppsala University in Sweden. And I'm really excited to be presenting today to this working group on metabarcoding. So thanks to Rob and the other organizers for this invitation to speak. So today I thought I'd give a very brief overview of long-read metabarcoding, how I've used it in the past, what are some other opportunities it offers, as well as some of the challenges with the method. And since I'm largely interested in the vast diversity of protists or microbial eukaryotes, most of my talk is going to focus on them, but the principles, of course, apply to prokaryotes as well. And of course, most of us, we're all familiar with regular short-read metabarcoding, which is a really powerful approach and where you're sequencing a short hypervariable marker, so usually less than 500 base pairs in length, to be able to sequence it on Illumina. And in protists, this typically tends to be the V4 or the V9 hypervariable region of the 18S gene. But of course, it can be other markers as well. But long-read metabarcoding, as the name implies, means sequencing longer barcodes. And these long reads, they can either correspond to one gene, like the 18S gene or the 16S gene, but it can also span several genes, such as the ribosomal RNA operon. And in this case, it would contain the 18S gene or the SSU gene, the ITS region, as well as the LSU gene. So the length of these fragments can be anywhere from, around 1,000 base pairs to several thousand base pairs. But in my research, I've mainly been using almost the entire ribosomal RNA operon as a barcode, which I amplify with general eukaryotic primers. So this gives an amplicon of about 4,000 to about 4,500 base pairs. And so what's nice with this method is that you get sequences containing multiple traditional barcodes, all on the same molecule, which gives you a really nice opportunity to directly compare them. Another advantage, of course, is that with long reads, you usually get more precise taxonomic classification, regardless of which method you're using. So you can classify more sequences down to lower taxonomic levels, simply because there are more nucleotides to tease apart closely related organisms. But perhaps more importantly for me, or more excitingly for me, these long reads have a much greater phylogenetic signal, and that allows you to build phylogenies, or at least more robust phylogenies of environmental diversity. And to illustrate this, I'm just going to show you two phylogenies of centrohelids, these very charismatic organisms so these phylogenies built from environmental sequencing data that I've generated. So the first is a phylogeny inferred from long-read metabarcoding data. So this is an 18S, 28S, so a two-gene phylogeny of centrohelids containing about 65 taxa. And there's two things I'd like to point out here. The first is that we recover the two main centrohelid groups, which is highlighted in the different colors. And the second is that this phylogeny is fairly well supported, and this is indicated by these circles. So the bigger the circle, that means the stronger the support for that branch. So next, I'm going to show you the phylogeny based on the V4 region. So I took the same 65 sequences, I trimmed them to just the V4 region to simulate short-read data, and I inferred another phylogeny. And this time, you don't recover the two main groups. They're not monophyletic, and overall, you have a less robust phylogeny. So this example really highlights that with long-read metabarcoding, we can get more robust phylogenies of the environmental diversity. And this is exciting for two reasons. So the first is that when you find novel diversity, which you almost always do, you can more confidently place it in the tree. And the second is that the method enables investigating ecological and evolutionary questions that rely on phylogenetic inference. So for instance, we used long-read metabarcoding data to investigate habitat transitions across the eukaryotic tree. And before I delve into the story, I do want to acknowledge all my co-authors and collaborators, in particular Fabian Berkey, with whom I planned most of this work, and Charlie Beaver and Daniel Vallot, who were really instrumental in processing short-read metabarcoding data. So habitat transitions, we're interested in them because they're major evolutionary events. They're very important drivers of evolution. They can trigger radiations. And one of the most difficult habitat boundaries to cross, and the one that we focused on, is the salt barrier. So this is the barrier between saline marine environments and the low salinity non-marine environments, such as soils and freshwater. And the salt barrier is very difficult to cross because adapting to this large shift in salinity is difficult, or at least that's one of the reasons why this barrier is hard to cross. But the question we asked is, how frequently have eukaryotes crossed the salt barrier? Are certain groups better at transitioning? And can we say in which habitat major eukaryotic clades arose? And to answer these questions, we started by collecting 21 samples from several different environments, including freshwater, soil, marine euphotic, and deep sea samples. So it's not, I mean, it's very limited sampling, but the aim here really was just to obtain a representative diversity for each environment. So we did long-read metabarcoding with general eukaryotic primers, and we got a fragment of around 4,500 base pairs in length, which we then sequenced on 4 smart cells of the PacBio Sequel 2 at the time. And I think this is maybe a good point or a good place to bring up this question that people often consider, which is, oh, should I be using PacBio or should I be using Nanopore for long-read metabarcoding? And the answer is, of course, that it depends. It depends on how long your barcode is. If it's short, shorter, like let's say up to 1 or 2 kilobase pairs, then I think either platform works well. But if it's a long barcode, say covering the ribosomal operon, I think I would still prefer PacBio because of its lower error rate. But it depends on other factors, such as cost and how many samples you have and how important portability is to you. And if you want to delve deeper, I really recommend this really great preprint that came out just a couple of months ago. Okay, but so from our 4 smart cells, we obtained roughly 10.7 million raw CCS sequences, which we then curated and clustered into nearly 17,000 OTUs at 97% similarity using the 18S gene. And we then performed phylogeny-aware taxonomic annotation of the 18S gene using the PR2 database as reference. And this way, you're essentially able to label the 18S part of the OTU. And because it's physically linked to the 28S molecule, we could transfer the annotation to the 28S gene as well. We then used both the 18S and 28S genes to construct phylogenies, and here is the tree that we got of the diversity in our 21 samples. So this phylogeny spans, or it contains almost all eukaryotic phyla and major lineages, which are represented by the different colors. For instance, here are the fungi, ciliates, radiolarians, and so on. Now, of course, for each of these OTUs, you know exactly where they came from. So when we mapped on the habitat data, we can visualize habitat preferences across the eukaryotic tree. So in this outer ring that you see, yellow indicates freshwater, orange is soil, and then the light blue and dark blue are the different are the sunlit and dark layers of the ocean. And essentially what you see is that the yellows and oranges are quite intermixed with each other. And that means that soil and freshwater lineages are quite closely related, and so are lineages residing in different layers of the ocean. But as we expected, we do see a very clear phylogenetic distinction between marine and non-marine lineages. So this confirms what previous studies have found. But of course, our tree is missing a large chunk of eukaryotic diversity. And so in an attempt to increase this covered diversity, we turn to short-read metabarcoding data. And, there's just taking advantage of the mass of available data sets that there are. So we included V4 sequences from the Malaspina expedition, from different forest and freshwater surveys. So in total, we got reads from 22 studies, and we clustered them into OTUs at a very coarse 97% similarity. We could then infer phylogenies, including the short read data, while using the pack biophylogenies that I showed earlier as backbone constraints. So this is a way to get around the lower phylogenetic signal of the short read data. But the nice thing with this is that we could then detect extra transitions that were missed by the pack bio data alone. And I'm just going to show you one example from the diatom tree. So here, all the branches or all the tips in orange are non-marine, like freshwater or soil taxa, whereas in blue you can see the marine taxa. And all of these blue marine, or this clade of marine taxa came from V4 sequences from ocean sampling day and Arctic cruises. So now that we had these more comprehensive phylogenies, we could ask questions such as, have certain groups more frequently crossed the salt barrier? And we could do this by modeling habitat transition in a Bayesian framework. So essentially, this is basically modeling the number of transitions per unit of branch length. So in this cartoon, the two colors show different habitats, marine and non-marine. And in this example, we see a single transition event or a low transition rate. Whereas on this phylogeny on the right, we observe multiple transitions or a higher transition rate. And so this way we can compare different clades and plot their habitat transition rates from low to high on the x-axis, with the y-axis showing the frequency. And now each colored curve here represents a different group. And what you see immediately is that transition rates do vary greatly across major eukaryotic clades. And that fungi have by far the highest transition rates, with at least 70 transition events detected, but we think this is probably a massive underestimate. And yeah, indeed, when you look at the fungal phylogenies, you can see that the marine and non-marine lineages are really closely related. This is followed by the cryptophytes and the ochrophytes. Next, we were interested in asking what the ancestral habitats of eukaryotic groups were. And we did this again by ancestral state reconstruction using in both maximum likelihood and Bayesian framework. And this figure summarizes our results. So at the tips of this tree are major eukaryotic phyla, and then the pie charts indicate the ancestral habitat for each phylum or major clade. So, for example, our analyses showed that metazoa likely originated in marine habitats, which, that's consistent with previous results. But we could do this for all of the different eukaryotic groups. But what I'd really like to point out is two of the largest monophyletic groups, called Amorphia and SAR. So Amorphia is the group that contains fungi, animals, amoebozoa, and other relatives. So based on the data we have and based on the models we used, this group likely arose in low salinity environments, whereas the SAR group, which includes diatoms, ciliates, radiolaria, dinoflagellates, etc., likely originated in marine habitats, meaning that the two largest eukaryotic assemblages seem to have arisen in different habitats, which is quite different from our notions of how life evolved. We tend to think all major groups arose in the sea and then colonized land. And while it will be interesting to see whether these results change with more data and with better models, I think it's really exciting that long-read metabarcoding offers the chance to even investigate such questions. Yeah, okay. But I guess like the main thing to take away is that the combination of long-read and short-read metabarcoding is really great for tackling evolutionary questions. And now I'm going to shift gears slightly because so far I've talked about just using the 18S and the 28S genes. But of course, long-read metabarcoding also gives you access to this fast-evolving ITS region, which could allow us to study biogeography at a much finer taxonomic resolution. And this is something I haven't looked at myself, but I do think this could be very useful. And just for this talk, I looked at some of my data to show an example to illustrate this. So I found, and this is just one example, but I found identical 18S sequences that you can see from this alignment corresponding to spumella or something like spumella from 6 different samples in 3 locations. And the 18S sequences, they seem to be quite abundant in all of these samples. So they were always found deep in the oceans, so either in the DCM, the mesopelagic, or the bathypelagic. So at the 18S level, these sequences are identical, but do we find differences in other genes? So I first looked at the 28S gene, also identical. And then I looked at the ITS region. And here I found that sequences, in this case, from the Mariana Trench in the Pacific, were 2 base pairs different in the ITS region from other sequences in the Indian Ocean. So the 2 populations could be slightly distinct. So this is just a tiny, tiny example based on very, very limited sampling, but I hope it illustrates how useful the approach can be. So long-read metabarcoding obviously has many advantages, but I did want to finish with some perspectives on challenges that are still associated with the method. The first thing to be aware of is that chimera formation rates may be higher in longer amplicons just because there is a greater chance for interruptions in the PCR or like premature terminations, which then leads to more incomplete amplicons that can act as primers in the next PCR cycle, and you therefore get more chimeras. So if possible, it's best to use as few PCR cycles as possible. But it's also worth adding that most most sequence processing tools like vSearch, USearch, Data2 now have chimera detection algorithms that are tailored to long-read amplicons. And then finally, I think we're still a little way away from standardized reproducible workflows that can generate ASVs from long-read metabarcoding data. So for example, the very popular NF-core AmpliSeq pipeline does not currently process nanopore data. That may change in the future as error rates get even lower. It is possible to process PacBio data, but in my experience, Data2 works well when your amplicons are, say, 1,500 base pairs, but not when they're 4,500 base pairs. Simply because to generate ASVs, you need a fraction of your reads to be error-free, and this becomes more difficult the longer your reads are. And you can also quickly run into memory issues. But that said, it is possible to generate ASVs, and there's a couple of studies which seem to suggest that USearch and vSearch tend to outperform Data2 for longer amplicons. But I think it still remains a challenge, especially for low abundance taxa. Right. So to conclude, despite some challenges, I think long-read metabarcoding continues to become more popular, especially as sequencing technologies become cheaper and have lower error rates. The longer amplicons mean more information for taxonomic annotation and a greater phylogenetic signal that can enable new research directions, more microevolutionary analyses. It can also offer access to the ITS region for organism groups that have traditionally used the 16S or the 18S as a barcode. And lastly, the technology is still new, and as it matures, we will hopefully see more efforts to standardize OTU and ASV generation pipelines. And with that, I'd like to thank you and everyone, all of my co-authors and collaborators on the study. Thanks.
Thanks so much, Mahwash. That was really great. Yeah, we have a few minutes for questions.
I'm going to try to stop sharing. Great.
Jed?
Hi, that was remarkable. I love that. One thing I'm curious about is we always find when we try to look at how universal a primer is to hit everything, it's very hard to compromise between that and getting the data that you want. And I'm curious if you've had a chance to try to look at the opposite of what you hit to see if you can figure out what you're not hitting with these primers. And I'm wondering if maybe some of those groups might affect that marine versus non-marine comparison, for example.
Right. I mean, yeah, I guess, even with long-read metabarcoding, you, of course, have the same challenges, many of the same challenges as with regular metabarcoding. There are definitely some groups that we missed. I would imagine the excavates. I'm trying to remember off the top of my head. But yeah, it's a lot of the fast-evolving groups, the usual suspects. And then for those groups, we didn't really, so I guess you're wondering about the ancestral state reconstruction where you're going further and further back in time. So maybe I can share my screen again, but essentially for those groups, we didn't really make predictions. Oops. Yeah. Yeah, so for instance, we missed a lot of these. I mean, it's hard to say. So some of these groups we missed just because of our sampling strategy. We didn't have shallow marine sediments, for example, So I think our sampling of anaerobic organisms was not that great. But then, yeah, we didn't really make so many predictions for those groups, to answer your question.
Okay, thank you very much.
I have a question about your annotation approach, right? So you mentioned just using the 18S region, right, and then extending that to your 28S. I guess, have you been making comparisons with with available 28S sequences or trying to build out a more comprehensive 18S to 28S database for reference?
We didn't really do a comparison per se. I think what we found, well, we were initially using the Silva database first, before we switched to the PR2. And I mean, overall, the 28S databases just don't have a lot of references. So it felt like 18S is the way to go. But yeah, I think it is a great way to build up 28S databases. Yeah. And hopefully, I mean, there is already, for example, Eukaryote, which is a database of across the ribosomal operon, and I think PR2 will also include the 28S gene at a later point.
There's a question in the chat from Juan. Says, I love the Juan, do you want to read your question or you
Yeah,
want me to?
sorry.
Yeah.
Yes, I love the result about the main eukaryotic clades. And you already mentioned that there are some limitation in the most divergent one, but I wonder if you take into account the closest prokaryotic, the closest relatives to eukaryote, the Asgararchia that are mainly saline and marine, and if that makes sense in the story.
Yeah, I think that's a tricky one. So we did originally consider it. Um, well, so we didn't include any prokaryotes at all. But we did try to we did try to use like all the proposed routes for the eukaryote tree. But I think as the reviewers very correctly pointed out, that was a bit of a stretch. Because these analyses, ancestral state reconstruction analyses, tend to be so influenced by these deep branching lineages. And of course, you have to take into account that a lot of diversity or a lot of lineages may have gone extinct. So, yeah, it's a bit hard to say.
Yeah,
Yeah.
it's super hard. Thanks.
Chad, do you have another question?
Yes, unless someone else does. When we were looking at the 18S data and analyzing the results, a lot of people are concerned about the copy number in the
Yeah.
18S. And I was surprised about the amazing stability you showed in your long reads, where you could find just two differences in the spumella of the Marianas Trench versus everywhere else. I would have expected even some within species changes in the ITS region that might be kind of random. So that was surprising to me how stable they were. And I'm wondering if you could comment on that.
That's a great point. I don't know I don't know so much about spumella. I don't know if anyone else has any idea of the copy number within ocrophytes in general. They were, because I did it just for this talk, and I I decided to not include, I don't know, look at amoebozoa or some other lineages where I do know they have a lot of copy number variants. But yeah, I think I was also a little bit surprised. But yeah, I will have another look, but just based on what I saw, yeah, uh, they did seem to be remarkably similar.
Yes, and in the ciliates, they even have a micronucleus and a macronucleus, and they make a lot of extra copies.
Yeah,
And
yeah.
that's really surprised me then.
Uh-huh. Yeah, and I think the dinoflagellates and radiolaria, they also are quite crazy. But it could also be that because I may accidentally have filtered out something because I was looking for identical sequences. So I started by looking at sequences that were 100% identical at the 18S level. So maybe that could be a reason why. But I do find it interesting that in all of these locations, these sequences seem to be quite abundant as well. Yeah.
Thank you.
Okay, great. I think in the interest of time, we'll wrap up there unless anybody has any other announcements. But thank you again to both of our speakers today. That was really, really great. And again, we're gonna try to schedule another one of these webinars in June. So be on the lookout for that announcement.